Hello, thank you for joining this webinar today. My name is Viviana Costa, and I'm a microcalorimetry application scientist here at TA Instruments. Today's webinar will focus on how our new high-throughput TA Instruments Rapid Screening-DSC can be used to help you accelerate your research. In the last few years, there's been quite an increase in the demand for developing new therapeutics based on monoclonal antibodies. Throughout antibody formulation development, we are seeing a trend where we see an increase in the desire for high concentration formulations to allow for smaller doses and make it easier for patients to self-administer these drugs. This is especially true for antibody formulations for subcutaneous and ocular drug delivery, where high concentrations are required to achieve the desired therapeutic effect. However, developing high concentration drugs is not simple, and there are several challenges such as stability issues and high viscosity. Because of this, it is important to have the right analytical tools that allow testing and characterizing this type of protein formulations at high concentrations. In today's webinar, our main focus is going to be on how we can measure thermal stability and why testing at high concentrations is important. One of the main characterization tests that is used in the biopharma is thermal stability testing. This is an important test because it allows us to predict a lot of factors like shelf life, the efficiency of a drug product, and also ultimately the quality of the drug. So how do biopharmaceutical scientists currently test for thermal stability? For this type of testing, there's really two techniques that are used: differential scanning calorimetry, or DSC, and nano-differential scanning fluorimetry, or DSF. And these are often used hand in hand. In simple terms, DSC measures the amount of heat that is consumed or released by a sample as it's heated, which means, for example, DSC can directly measure the heat that is absorbed as a protein unfolds. On the other hand, DSF measures changes in intrinsic protein fluorescence as the protein unfolds, or in other words, DSC measures changes in fluorescence rather than in the protein's melting point. Because of these differences between these two techniques, in general, researchers often say they like DSC better because it's the gold standard, and it also gives great data. But it does have some drawbacks in terms of throughput, sample amount, and the need for sample dilution prior to measurement. On the other hand, DSF has great throughput, but data is not as information dense as the data you get from a DSC. And it can also lack resolution, so you might be missing subtle changes or shoulders in the thermogram. So is there a better way? Yes, there is a better way. And this is where the new Rapid Screening-DSC fits in very nicely, because we have addressed the challenges of dilution, sample concentration, cleaning, and throughput needs to meet our users' challenges, all while remaining very high quality data that we are used to for DSC. So let's take a closer look at some of the key features of RS-DSC. One of its greatest features is the flexibility in throughput, where we can analyze up to 24 samples at the same time, which really redefines the thermal stability testing landscape, where for the first time, we are providing high throughput DSC thermal stability testing. This is going to really help you expedite the development and characterization of the thermal stability of drug products. Another great feature of the RS-DSC is that it's dilution-free. This is only possible because this instrument is uniquely designed to handle high concentration biological drug formulations by using microfluidic chips, which is something we'll be discussing later on in this webinar. So by being dilution-free, it allows scientists to test at the final formulation strength concentrations. In addition, testing at high concentrations is also going to provide you additional new insights that we previously didn't have access to. Now, this is another key feature that I think a lot of you will appreciate. With the RS-DSC, there is no cleaning that is required. And that's because we are using disposable microfluidic chips. This is going to eliminate the need for repetitive cleaning of the instrument between runs, which means it's going to save you time, but also it's going to reduce the risk of cross-contamination. So basically, a sample is prepared and sealed, and it's ready for analysis in less than 1 minute, requiring only minimal amount of volumes, which is 11 microliters. Finally, we are also introducing new nano-analyze software features that, for example, are going to allow you for automated baseline construction and detection of the unfolding midpoint temperatures. And you can also be able to compare data from up to 96 thermograms. Before we discuss some of the uses and applications for the RS-DSC, let's take a quick look at what happens to a protein during a DSC scan. Earlier in this webinar, I mentioned that a DSC measures the differences in heat flow between a sample and a reference material. So by looking at the difference in heat flow between the sample and reference, DSC is able to measure the amount of heat that is consumed or released during a transition. So in this example here on the right of the slide, we are looking at a DSC thermogram to study protein unfolding. Here, a solution of protein is going to be heated at a constant rate in the calorimeter alongside a reference. As the temperature in the cell increases, heat is going to be absorbed by the protein, which causes the protein to unfold, and this gives rise to an endothermic peak, as you see here in this graph. The unfolding happens over temperature range, and this temperature range is characteristic for that protein. The max of this temperature range corresponds to the melting temperature, or TM, which means we can use DSC as a tool to characterize protein stability, since there is a direct correlation with unfolding temperature and stability of the biomolecule. So what type of thermodynamic data can we directly extract from DSC that is relevant for studying biomolecule stability? So a DSC thermogram is going to give us the T onset, which is the temperature at which a biomolecule begins to unfold. It also gives us the midpoint temperature of that transition, or TM, which is the point at which 50% of protein molecules are on average unfolded. The shape and area of the transition endotherm also contains thermodynamic information about the process. So the integrated area under the peak in this example, in this DSC trace, divided by the total amount of protein gives us the enthalpy for the unfolding transition. So in this example, we are looking at replicate scans for a monoclonal antibody at 100 mg per ml. You also notice how TM values are very reproducible, which is really a characteristic of the great quality data you can get from a DSC. And finally, I also want to mention that DSC data also can indirectly provide values for entropy and gives free energy. Understanding the structure-function relationship of antibodies is fundamental for comprehending how functional variations arise, whether due to alterations in solution conditions or sequence modification. So by measuring the short-term stability under different conditions, one can have a better understanding of these drug products. So for example, we can look at formulation stability by screening for biomolecules more likely to undergo denaturation or degradation. We can also look at formation of higher order structures, which may happen at higher concentrations. It can also be useful for candidate selection for understanding mutations in our protein and how they are affecting this protein stability. We can also look at and compare biosimilars to get insights into new modalities and chemistries, and of course use thermal stability measurements to ensure quality control with no batch-to-batch variation. This is an introduction slide to the modalities that can be studied using DSC. And these are proteins, nucleic acids, lipids. However, in today's presentation, we are mostly focused on protein applications. One of the most important questions during drug development is what is the short-term stability of a biomolecule? This can be understood by measuring TM using DSC, since again, DSC is the gold standard for measuring melting temperatures. So every protein is going to have a unique DSC thermogram. And things like buffer, pH, excipients can affect the stability and unfolding profile of a protein. So DSC can be used to evaluate that. Antibody candidates, for antibody candidates, the general consensus from the scientific community is that their optimal thermal properties in terms of stability should be a T onset greater than 55 Celsius and a FAV TM greater than 65 Celsius. So in this example that we show here, we can see this antibody unfolds in 3 transitions with some overlapping, but using our analysis software, it is possible to deconvolute these multiple overlapping transitions. For this antibody molecule, the first transition is associated with the CH2 domain and has a T onset of around 66 Celsius. The second transition corresponds to the Fab domain, with a TM of about 78 Celsius. And finally, the CH3 domain unfolds last with a TM of about 85 Celsius. So one could conclude that this antibody meets the minimal thermal stability criteria and could therefore advance on to the next formulation development step. In this example, again, we look at the thermal stability of antibody candidates, but here we are comparing DSC thermograms from different types of antibodies. In this case, they all have the same constant region, but they have differences in their binding region, which translates to differences in the DSC traces, as you can see here in this graph. Here we are seeing differences, that differences in the binding region is going to affect the overall short-term stability of these antibodies. So again, this observation can be used to, for example, guide you through candidate selection. Optimizing protein structure and function is essential in drug development and protein development. So things like protein mutations, single amino acid modifications are a common strategy that is used to improve protein stability. So in this example, we are looking at how antibody stability is affected by sequence modifications in the case of engineered antibodies. So here we see the parent antibody with an unfolding of the CH2, Fab, and CH3 domains that is contained within a major thermal transition with a Tmax of about 76 Celsius. For the parent protein, a single amino acid mutation, seen here as mutation 1 in blue, was made, which had no major effects in terms of short-term stability of the antibody. However, additional single amino acid mutations did have a significant impact on the protein stability, as seen here in the case of mutation 2 and mutation 3. So, for instance, the presence of an obvious shoulder is observed in mutation 2, which likely corresponds to the destabilization in the CH2 domain with an added Tmax 1 at 70 Celsius. This is going to be coupled with a small shift in the main transition at a lower temperature relative to the parent protein. In the case of mutation 3, you can see that it causes significant destabilization of the CH2 domain, as you've seen here by the presence of an additional unfolding peak with a Tmax 1 of about 65 Celsius. So with DSC, we can compare the structural stability of the protein with different sequence modifications and select the most promising ones to move forward in the development process. In this example, we are looking at triplicate data for 20 mg per ml trastuzumab formulations in different buffer conditions. The first unfolding event, corresponding to the unfolding of the CH2 domain, is not significantly impacted by histidine, borate, and PBS buffer. However, the succinate buffer does destabilize the CH2 domain, resulting in a decrease to lower temperatures of the T onset of unfolding, and also Tmax by about 3 degrees Celsius. With respect to the main transition, which reflects the Fab and CH3 unfolding events, the histidine and succinate buffers are the most stabilizing, with a Tmax of about 82.6 Celsius. The main transition, in this case, is going to be less stable in the borate buffer, with a Tmax of about 69 Celsius. So overall, one can conclude that the most stabilizing buffer formulation for this antibody is histidine buffer, which is actually the buffer that is used for the final formulation of the approved drug product. There's also an increased interest in making high concentration dosage forms. So another question you might have is if protein concentration affects its stability. We have seen that formulating proteins at high concentrations can alter their thermal stability. In some cases, it has been shown to decrease stability, while in other cases, it has been shown to improve the thermal stability. So it is important to be able to evaluate and determine melting temperatures at high concentrations. Until recently, testing proteins at high concentrations was difficult because the technology did not allow for this. However, with our new RS-DSC, you can test proteins at high concentrations. And this is what we show here in this graph, where we are testing lysozyme at various concentrations. As the concentration of lysozyme increases from 30 to 160 mg per ml, you can see that Tmax shifts to higher temperatures, as can be seen in the inset figure, where we see a linear increase in Tmax as a function of concentration. However, as the concentration keeps increasing to around 280 mg per ml, the thermal stability plateaus at a Tmax of about 69 Celsius. Now, this is not throughout all the concentration range. So at concentration of about 300 mg per ml, the Tmax shifts to lower temperatures, meaning lysozyme now is less stable at these higher concentrations. In addition, you can also see the presence of a shoulder at the tail of the main transition at these higher concentrations, which most likely is related to the formation of higher order structures at higher concentration. So you can see here that with the RS-DSC, we are able to test the very high concentrations and see how concentration affects the protein stability. This is another example where we are looking at chymotrypsin in PBS buffer and how its stability is affected by concentration. Again, here we see a dependence of the main protein transition Tmax's as a function of concentration. So in this case, there's actually a decrease in stability as you increase the protein concentration in PBS, which is especially noticeable when you go from 20 to 60 mg per ml. This data again highlights the importance of evaluating the drug at its final concentration of interest, since testing in dilute samples might not reveal the actual thermal stability, as there might be a stability dependence with concentration. A special class of antibody therapies that you see a lot in development are antibody drug conjugates, or ADCs, which are an antibody with at least one small molecule attached via a linker. So obviously, one question you might have during ADC development is if conjugating a drug will affect the protein's stability. So this is something that makes sense, right? Because attaching a small molecule to the antibody can cause changes in the overall structure of the antibody and therefore affect their stability. So how do we evaluate if attaching a small molecule affects the antibody structure? We can use DSC for that. So in this example shown here, an antibody is run in its native form without any modifications, and then also with different loads of a drug conjugate. So you can see the unfolding of the native antibody displays one large asymmetrical peak, which is going to be attributed to the unfolding of CH2 and Fab domains. And this transition is going to be followed by a smaller transition associated with the CH3 domain. When looking at the thermogram of the different drug loads, we can see that the presence of a new transition which is increasing as the magnitude of the drug load increases. So this indicates that the attachment of this small molecule, it's destabilizing a portion of the CH2 domain. And we still have a portion of the CH2 domain unfolding with the Fab domain, as well as there's still some asymmetry in the main peak transition, but we have also a lower enthalpy. So there's part of it that is still unfolding with Fab, but most of it is unfolding at lower temperatures. Now, you also notice that the CH3 domain is more or less unaffected by the linkage of the small molecule. This is another example with an antibody drug conjugate. In this case, we are looking at the effect of different payloads as well as formulation buffer on the stability of the antibody. So here the antibody, which is trastuzumab, is run without any modifications, which we label parent, and then also with two different drug conjugates, which we labeling ADC1 and ADC2. You can see in this data that in the parent antibody, the succinate buffer relative to PBS buffer notably destabilizes the first transition, which corresponds to the unfolding of the CH2 domain. On the other hand, succinate stabilizes the second transition, which is related to Fab and CH3 domain. Now, looking at ADC1, you'll notice that the CH2 domain is destabilized significantly very much relative to the parent antibody, but remains relatively unchanged between different buffer formulations. The second transition also sees a slight decrease in stability for both buffers relative to the parent antibody. Finally, for the second ADC or ADC2, the first transition related to the CH2 domain is destabilized in PBS buffer relative to the parent antibody, but is also stabilized when switched to succinate buffer. Like in the case of ADC1, you'll also see that the second transition in ADC2 sees a slight decrease in stability for both buffers relative to the parent antibody. So far we have looked at proteins and antibody thermograms, so let's take a look at another type of biomolecule. And this is going to be our final application example. So here we show two different FDA-approved oligonucleotide duplexes. When working with duplexes, you'll see an increase in TM as strand concentration increases, which is what we observe for both samples shown here. Furthermore, you also see that as the duplex length increases, melting temperatures shift to higher temperatures independently of concentration. So again, we show the value of testing at higher concentrations. And in this case, we went as high as 140 mg per ml for oligo solutions. So one can evaluate how concentration affects stability of these biomolecules. After discussing all the previous application examples, we can for sure appreciate how important it is to understand biomolecule stability under different conditions and at the required high concentrations to support drug development and ensure quality data. This is why we designed the RS-DSC instrument to meet this demand for a rapid thermal stability characterization for high concentration biotherapeutics. In this slide, we summarize some of the groundbreaking capabilities of the RS-DSC. So here, we already discussed a lot about these capabilities, but we can see that the RS-DSC has the ability for testing an extensive range of sample concentrations without the need for dilution, as well as the capacity for analyzing very high concentration drug products in an efficient and effective way. Testing is also done with minimal sample volume requirements to help you minimize material use and cost. And finally, it also simplifies testing by removing the need for cleaning. In this slide, we'll be looking at how the workflow for the RS-DSC looks like and how simple it is to prepare microfluidic chips. So what you'll see here is that we have this disposable trays of microfluidic chips that are set up for 24 samples. You can run as many as you want, and you can keep any that you don't use for later. Here we are filling 11 microliters of sample into the microfluidic chip using a pipette. Next, to prevent against evaporation of sample, we take an adhesive cover slip from the cover slip release assembly tray and place it on top of the microfluidic chip and gently press. Next, you place the sample microfluidic chip into the sample side of the calorimeter. The reference chips are on the right side, and these stay the same every time. Next, you need to cover the calorimeter with these inner lids that hold the chips down during a scan, and then close the latch of the instrument's lid. So basically, this is the full workflow for the RS-DSC. This is just a small summary overview of what the microfluidic chip preparation looks like, as we just saw in the previous video. Again, all the microfluidic chip preparation tools are designed for you to easily prepare samples. All the tools you see here have alignment features to help you place the cover slips correctly on the top of the chip. Because these are disposable chips, after you run your samples, the microfluidic chips are thrown away, therefore completely eliminating the need for any cleaning of the instrument. This is just a close-up of the calorimeters in the RS-DSC. The important thing here is just to notice that each calorimeter is an independent unit with a reference and a sample side. Where the microfluidic chips are placed is on the left side, and on the right side you'll see the reference peak chips in the calorimeter. The microfluidic chips are replaced in each run, but the reference peak chips are kept in the calorimeter. Finally, I also want to highlight the automated features in our data analysis software platform, NanoAnalyze, that makes DSC data analysis and processing much easier for the user. These features are powerful, and they help address some of the pain points related to the data processing, like how time-consuming it can be, as well as removing any subjectivity when drawing a baseline. So the way that this works is that within the software, you're going to be able to import up to 96 scans at the same time into a function called Rapid DSC. At this point, you'll see a similar window as you see here in the slide. So here the software allows you, for example, to select temperature range for where you want to look for transitions. It also allows you to select how many peaks, and you can select up to 3 that you want to look for. So with just a quick press of a button, the software will process through all the scans that you imported. And it's going to process this automatically. It's going to automatically fit a polynomial baseline to give you values for the temperature max for selected transitions. If you want to look at more details for each of the data sets, you can click on the little graph icon that you can see here, which will open a pop-up window that looks like the one in this slide. Within this window, a user can take a closer look at the data, or maybe if they want to edit the baseline to make sure that the Tmax is where it should be, the user can also do that. So there's still a lot of user input flexibility within the automated features of NanoAnalyze. Finally, I also want to highlight some other great features like the ability to make high-quality overlay graphs or Tmax visualization graphs that are going to help you interpret larger data sets. For instance, it's going to be easier to find outliers or maybe quickly find higher stability candidates. This is just a summary slide of what TA Instruments has to offer in terms of biomolecule characterizations. In addition to what you just saw here today for our RS-DSC instrument, here you can look at each of these instruments and their various applications across the drug development process. So I will recommend visiting our website and take a look at our full portfolio. So very quickly here, we have just a summary slide of some of our great software capabilities, as well as some details as it relates to service support. There are also a lot of application resources in our website, and I would encourage you to go there. Here is a link at the bottom that you can use where you'll find a wealth of information to support biomolecule development. If you'd like to learn more about our biomolecular characterization capabilities, please visit this link here. And that's basically all I have for you today. So thank you so much for your attention. And if anybody has any questions, I'll be more than happy to take them.
Hello, my name is Neil DeMars, and I'm the Solutions Product Manager at TA Instruments. Thank you for attending today to learn about our new high-throughput DSC technology, the RS-DSC. At this time, we're happy to answer any of your questions, and I already see that we have a few in the queue. Please continue asking your questions, and we'll get to them. Let's put it here. So first question, Viviana. I'm used to using a matching buffer for the reference cell, but here I see there is a peak chip. Why is a buffer reference no longer needed?
Thank you for that question. Yes, there is no need for a buffer reference. So the primary function of the reference in any DSC instrument is to match the differential thermal response of the sample to any transient noise. So obviously, having a reference with a similar heat capacity helps to subtract out events that are not related to your sample. However, in the RS-DSC, the majority of the heat capacity of the sample side actually comes from the glass microfluidic chip. So because this chip is much larger than the liquid sample volume. So what this means is that you can easily match the heat capacity from the sample cell with just a peak chip, which is what we use in the RS-DSC.
Thanks. All right, second question here. How does DSC data compare to other stability testing methods like nano-DSF?
Sure. So in the case of DSF, data can be difficult to deconvolute, especially when you have multiple transitions. So it can be, let's say, difficult to determine accurately multiple melting points during unfolding. You can also have loss of resolution for smaller magnitude transitions. So in this sense, DSC is a great technique because it does provide you great resolution. So it's easy to deconvolute and identify multiple peaks. And of course, you also get more information out of DSC experiment since it's also measuring enthalpy. Plus, you can also use it for different modalities without any concerns for like molecule intrinsic fluorescence, for example.
Here we go. Somebody's. Okay. Next question. Will any of the sample chip prep be automated in the future?
That's a very good question. So sample preparation that we have for the RS-DSC is very simple. It does not take that much time to prepare a set of 24 microfluidic chips, but it does still take time. So we want to improve testing workflow. So it's something that we are always looking to add in the future. So definitely we are addressing, including automation in sometime in the future.
Looks like we might have a previous DSC user here. Do you need to degas samples?
Typically, it's best practice to degas samples for DSC analysis in general. And obviously, this is because as the solution is heated, any gas bubbles that might form during the scan can cause random thermal events in the baseline. In the RS-DSC, since the volume is much smaller, you might not find the need to degas, but we do still recommend just as a best practice.
Okay. Do you need to run 24 samples, or can you run less than the RS-DSC?
You can run as many as you want, so you don't have to fully run 24 samples in one experiment. So one up to 24 will be your choice.
Got a couple more questions, but it looks like these are kind of addressed these. Is there a need to run a a buffer solution for background subtraction. I think we answered that. And then the final one, do you need to degas the samples? So I guess we can give another couple seconds if there's any last minute questions. We're happy to answer those. Otherwise, you will have access to this. Everybody who registered should receive a link to view this in the future. Just got a question popped in. Thanks so much. Let's see, are the chips reusable?
No, they are disposable. So once you use them you're just going to dispose them at the end of the experiment. So, and this is great because what it means is there's no cleaning that is involved when running experiments in the RS-DSC.
Okay. Yep. So, all right, well, thank you, everyone. If you have questions that come up in the future, please reach out to us directly or your local representative. We're happy to talk to you about our RS-DSC or any of our other equipment that we provide for you. Thank you for your time.